We're so excited to have Jillian, is it Coles or Cowles?
Coles,
Coles. Join us all the way from Arizona this evening to enlighten us and teach us all about the amazing world of arachnids. And Jillian is a retired microbiologist, but she has spent a lot of time researching, studying, and photographing arachnids. And in 2018, had an amazing book published by the Princeton University Press called Amazing Arachnids. Jillian, thank you so much for coming and sharing your knowledge and expertise with us this evening.
Well, thanks for having me. Should I go ahead and share screen now?
Go ahead.
Great. Okay.
One thing before Jillian gets started, questions are highly encouraged. Put your questions in the chat box, but we are going to wait until the end of the presentation to kind of take all the questions at once then, okay? But put them in the chat box as they come in. Your mind.
Let's see, I'm trying to get it to the slideshow. I can't get this bar out of the way.
Right over there to the big, it says from the beginning.
Just a moment. This bar is in the way for me to click on slideshow. I need to, maybe this will work. Start from beginning. Let's try that. Yeah. Okay. All right. So whenever you want, I can start. Can everyone see this slideshow now?
We can.
Okay, great. And you can hear me okay?
Broadcasting loud and clear.
All right, excellent. So the title of this talk is called Sharing, Caring, and Thievery, Arachnid Behavior and Interactions. So let's start right off with what is an arachnid? Well, first of all, it's an arthropod, which means it has an exoskeleton. That also means it has to molt in order to grow. And it has multi-segmented legs and body. So among the arthropods are insects, crustaceans, which include things like sow bugs, centipedes, millipedes, and chelicerates. Now, among the chelicerates are horseshoe crabs, arachnids, and possibly sea spiders, although that's a little bit controversial. And the structure that gives chelicerates their identity are these little structures right here. In a scorpion, each chelicera has a fixed and movable finger. So in a scorpion, they use these like little hands for cutting up their food. For example, this is a katydid that this scorpion is masticating and introducing digestive enzymes into, and then it'll slurp down the pre-digested liquefied food. So each little chelicera has a fixed and movable finger, and it can cut up its food. Whoops, flipped a little too fast. So they're almost like appendages, like little extra legs. And from a developmental standpoint, they are like little extra legs. But from a functional standpoint, they're more like jaws. So in spiders, each chelicera has a single naked fang. And these fangs are hollow inside and have a little hole at the tip so that they can inject venom. So among the arachnids are spiders, and this is the spider holding the butterfly, okay. Scorpions, pseudoscorpions, Europigids, also known as vinegaroons, schizomids, amblypigids, also known as tailless whip spiders, palpigrades, which are also known as micro whip scorpions. Now, they're not a scorpion. These things are only about 1 millimeter long, including the flagellum. So these are very tiny little animals that can actually live within the spaces within soil. Harvestmen, sun spiders, also known as solifuges or camel spiders. Riccinuleas, also known as hooded tick spiders. And the mites and ticks. Now, mites and ticks used to be in a single group, but in more recent time, they've been split out into two separate groups. One is the Parasitoformes that includes ticks, Opilioacarids, and a few other special kinds of mites. And the other group is Cariformes, which includes red spider mites, giant desert velvet mites, and that sort of thing. Mites that are most people think of as more typical mites. So let's get started with green lynx spiders. These are a group of species that are found throughout the tropics and the subtropics. There are many different species of them, but they all are ambush hunters, and they depend a lot on their eyesight for capturing prey. In this case, this green lynx spider was lying in wait for pollinators to come visit this flower, and it captured this little bombyliid fly simply by pouncing on the fly and grabbing it and then biting it. So these are very commonly found on flowers like prickly pear flowers in the spring in Arizona. And we have two species of green lynx spiders in Arizona. One is Pseudoscorpions viridans, which has a highly variable color in the abdomen. And the other one is Pseudoscorpions longipalpus, which is mostly green. Now, here's a green lynx spider guarding an egg sac. And because these spiders have evolved in tropical areas, they have to guard their egg sacs against all sorts of other insects, such as ants, grasshoppers, katydids, and other animals that would very happily chow down on these eggs. So the mother green lynx spider guards her egg sac against insects, but she also guards her egg sac against vertebrate predators. So when a green lynx spider sees a threat approaching, what she'll first of all do is face the threat. And you'll notice at this point in time, the bottom of her body here, from her abdomen across the bottom of her cephalothorax, is more or less a straight line. But as the threat approaches, which is this pair of forceps, she tips her cephalothorax upwards a little bit. So the front of her body is the cephalothorax, and she tips it up a little bit, and she extends her fangs forward a little bit. And then she spreads them apart, and she actually can squirt venom out of her fangs for a distance of several centimeters. And it comes out in a fine stream. Now, one of the fangs has already ejected, and you can see the droplets on this forceps. So it's quite a bit of venom. And here's the other fang still squirting out the stream of venom. In this particular instance, the spider was trying to pull free from the forceps. You can see her leg actually moving here. It's not being actually held. It's just loosely touching her leg, but she's pulling free, and her aim was thrown off a little bit. You can see the stream of venom right here. And in this case, she's actually sitting right on top of her egg sac and squirting the forceps as they approach. Now, the first time that this was ever documented was back in the 1940s. And right after World War II, there was a serviceman who was checking his motorcycle, and he knelt down behind the motorcycle and was checking the tire or something. And all of a sudden he felt an intense pain in one of his eyes. And he actually had to go and get medical treatment of his eye. It was so bad he couldn't see out of it for a couple of days. And the doctor was very curious about what had done this to his eye. So the doctor had them investigate behind the motorcycle. And I guess at the license plate or somewhere at the back of the motorcycle, they found a female green lynx spider hiding with her egg sac. And she apparently saw the man, you know, just kneeling and looking at the back of the motorcycle, and she squirted him right in the eye with the venom. So this is just a closer view of this spider actually ejecting venom. So the female spider hangs out near her egg sac until the babies have emerged from the egg sac. And then they undergo a molt a few days after they emerge. And here's the little exoskeletons. As I mentioned, arthropods have to molt in order to grow. So they molt before they leave. And then they stand on their little tippy toes on a nice breezy day, and they release silk out of their rear end, and they actually balloon away to a new life. If the mother spider was removed from guarding her egg sac, the success rate of that egg sac goes from roughly 70% down to about 10% because of predation of that egg sac, which would include things like insects as well as mice and possibly birds. So guarding an egg sac is one of the most common kinds of maternal care of young that you see in spiders, although only the green lynx spiders are known to squirt venom as a defense mechanism for guarding their egg sacs. Now let's look at another arachnid, a vinegaroon. So vinegaroons can squirt almost pure acetic acid. It's like 85% pure acetic acid out of their rear end as a defensive mechanism. And they squirt it out of this little structure that's called a pygidium, which works like a revolving gun turret. And they can actually squirt in any direction with this. This spray is used strictly for defensive capabilities, capacities. It is not used for capturing prey. They use these heavy pedipalps for capturing prey. Now, vinegaroons undergo a very, very lengthy courtship that involves a dance stage. The male on this side is holding the antenniform legs of the female in his chelicerae. So the antenniform legs of the male, this is a male antenniform leg here. They are legs that have evolved to the point where they're used purely for sensory and for communication, not for locomotion anymore. But he holds the female's antenniform legs in his chelicerae, and he nibbles on them as they do this dance. And the dance where they pull each other back and forth can last for several hours, like 5 hours. There he is holding the female's chelicerae in his, the female's antenniform legs in his chelicerae. And then after the male determines that, that he's ready to generate a spermatophore, the female stands behind him, embracing his abdomen. And for the next 3 hours, he has to produce a spermatophore, which he then deposits on the substrate, and she picks it up with her gonopore. And then he turns around and embraces her abdomen while he pushes in the contents of the spermatophore. Now that entire courtship can take 13 hours or even longer. So it's a tremendous investment in energy on the part of the vinegaroons when they court and mate, but that's just the start, because that happens in late summer. And in the fall, the female digs a maternity burrow where she seals herself underground, and she'll stay there for at least the next 7 months. And in the spring, she produces eggs that she deposits into this clear membranous pouch that she holds on the underside of her abdomen. And I call this a belly bag. And the eggs incubate for about 5 weeks. And then when the babies hatch, they climb onto the mother's abdomen, and they cling there for another 5 weeks while they undergo further development. And they tap into food reserves in these fat little bodies. They don't feed on anything during that time. They just simply use these food stores that they were born with. And they hold onto their mother's abdomen with these little sucker pads on the tips of their legs. Sometimes a mother produces her own weight in babies. The average number of babies that a wild vinegaroon mother produces is somewhere around 52 babies. at a time. So it actually can exceed her weight. At the end of 5 weeks of development, the babies molt and they drop to the ground. They had to free themselves from their old exoskeletons that were, once again, clinging to the mother's abdomen with those little sucker pads. So they free themselves from their old exoskeletons, drop to the floor, and at that point, they're colorless. But over the next few weeks, their pedipalps will turn first pink and then red, and their bodies turn from white to eventually black as their exoskeleton hardens. So at the end of about 4 weeks, they look like this. At that point in time, sometimes a mother will actually exit the burrow along about July, and she sometimes will capture prey and bring it back and deposit it in the burrow for the babies. And in this case, she had captured a large cricket and had deposited it, and the babies all swarmed around the cricket and were tearing it apart in the burrow. Once the babies leave the burrow, the mother continues to share food with them. And in captivity, we've seen the adult females continue to share food for as much as 4 months after the babies emerge from the burrow. And in this case, this mother Vinegaroon had not fed for a good 7 months. She had produced almost her own weight in babies and was absolutely famished when she emerged from the burrow. And her very first meal, this cricket, she was sharing with her babies. Here's a little baby reaching right between her pedipalps to grab chunks of cricket to eat. And sometimes the babies actually swarm around the mother and just climb on top and grab chunks. Here's one with a chunk, here's one with a chunk. So as she masticates that cricket and makes it nice and soft and easy to tear apart, the babies can pull off chunks and run off with them. So the first time I ever saw this, I was really blown away because, like most people, I assumed that extended maternal care of young is pretty much in the zone of warm-blooded creatures such as birds and mammals. And I didn't expect an arachnid to show this kind of extended maternal care of young. But when you think about it, any benefit that she gives these tiny little baby vinegaroons to grow into the next size larger, so that they can get past this very vulnerable stage, will increase the chances of her genes being passed on to the next generation. So there's a selective advantage in altruism on the part of the adult females to share food with their offspring, just as there is for humans to help their offspring survive. The same principles of natural selection apply to arachnids as do to humans. So let's look at another kind of arachnid that shows maternal care of young. This is a cousin of the black widow. It's a tiny little spider called Enolosimos arizona. And these particular spiders form colonies that are founded by a single mother. And all of her offspring live in this one colony. Now, to give you some idea of her size, this is the adult mother, and she's bringing a fruit fly to share with her babies. So here she's brought a fruit fly to this batch of babies and this batch of babies, and she's allowing them to feed on the fruit flies without her eating any of them. As the siblings, as the offspring grow larger, they start to capture prey themselves in the web. And so if one of them finds prey that's too large to tackle by themselves, they signal to their siblings to come and help. And they cooperatively capture prey and then share in the prey. So these Enolosimos arizona are called subsocial spiders because they will disperse as they reach sexual maturity. So the maximum size of any one colony is only the offspring of a single female spider, which is only about 30 spiders total. But in the tropics, there are other species of Enolosimos that have permanent colonies that run year-round, and have multiple generations. And those colonies can reach as many as 50,000 spiders in a colony. Now, here's something of interest also living in the web. This is a kleptoparasitic mirrored bug. This is a little nymph of the mirrored bug, but the adults also do this. They live in the webs of these Enolosimos, and they actually feed on the prey. Here, this same fly that those spiders were feeding on is being tapped into by this mirrored bug. And it's a kleptoparasitic bug that lives in the webs of these subsocial spiders. So far we've talked about maternal care of young, extended maternal care of young, and subsocial spiders. And all of those are predicated on maternal care. In other words, it's all mothers and babies that are sort of the foundation of the social behavior. Now let's talk about some social behavior that is not predicated on maternal care. This is a little dictinid spider, and dictinids are specialists on flies. So they built their webs. By the way, this is the same fly as in the next photo to give you scale. That's the same fly. So these webs are really tiny. They can be at the tips of little shrubs or at the tips of grasses. For example, in this grass seed head, if you were to look at the base of the grass seed head, turn around and look there, you'd see these two little dictinid spiders. There's the female and there's the male. And what these spiders will do is cooperatively kill prey together. So it's a mated pair that will cooperatively kill prey. This is Dictina calcarata, a different species of dictinid. And this is a male, and I'm gonna point out this pointy little pedipalp that he has because it'll help you pick out the male in future pictures. So one day I was photographing some bombyliid flies that were sunning on a boulder, and this particular species of dictinid really loves to build webs on sunny boulders where flies will go and sun themselves. They also like sunny windowsills. So here's the male spider in the background. You can see that little pointy pedipalp again, and here's the female, and they are cooperatively killing this fly. And they could barely overcome the fly. I mean, these are tiny little spiders. They're only about 3 millimeters long. And this fly was putting up quite a struggle. And together they were able to capture the fly. And then they both settled in it and shared in the feast after killing the fly together. Another pair of spiders that do sharing of food are the cellar spiders. So cellar spiders are frequently seen in closets or cellars or garages. Some people call them daddy long legs. But this pair of cellar spiders, the male had captured this Centauroides scorpion in my bathroom. And the female came over and she signaled that she was interested in feeding on this, and the male allowed her to share the feast. And they actually fed on it together for 24 hours. So this chivalrous behavior probably benefits the male cellar spider. It probably gives him some preferential treatment when it comes to breeding rights to that female. So far we've looked at all kinds of interactions between animals within a single species, but sometimes arachnids interact with other species. For example, these are some mites that are phoretic mites on this carrion beetle. So carrion beetles are really interesting because what they do is they'll form a pair bond, and when they find a little carcass somewhere, such as a little rodent or a little bird, they'll bury the carcass, and the female carrion beetle will lay an egg or a couple eggs in that little carcass. And so they work together to bury the carcass and lay the egg in it. Now, these mites on this beetle are not feeding on the beetle at all. They're simply riding on the beetle, using it as transportation. And when I offered this beetle some carrion in the form of moist cat food, the beetle immediately started feeding on the carrion, and all the little mites dove headfirst into the carrion and were also feeding on it. But as the beetle finished feeding, all the mites climbed back on board and not a single mite was left behind. Now you wonder, why would the carrion beetle be carrying around these mites if the mites are also feeding on carrion? Well, when a male and female carrion beetle bury a little carcass and lay one of their own eggs in there, what these little mites will do is dismount. They'll mature to fully mature mites. Right now, they're just immature mites. They'll go ahead and mature, they'll mate, and then they'll lay their eggs in that carrion also. Now you wonder, are they competing with the beetle for that resource? No, they think that what those mite larvae, what those young mites will be doing is killing fly maggots and other competitors for that carrion. And they might actually increase the chances of the carrion beetle's offspring being able to have better access to that resource. Of course, a little carcass is a very ephemeral resource. It's not present for very long, and it's also spatially very rare. So there's no way that these little mites could get to the carcass without the transportation of the beetle. But in return, they probably repay the favor by eliminating competitors that might compete with the beetle's larva for that resource. So I mentioned Fluoresce, which is hitchhiking, and the little arachnids that are most famous for Fluoresce are Pseudoscorpions. So this little pseudoscorpion is only about 3 millimeters long. They're quite tiny. And what they're famous for is grabbing rides on other arthropods. Now, this particular pseudoscorpion is grabbing the leg of a Stratiameid fly. This is a soldier fly that is getting a little drink of water near the edge of a wash. And this little pseudoscorpion is getting transportation probably from one rotting cactus to another rotting cactus eventually, where there's lots of little arthropods feeding on that rotting cactus. But this Stratiameid fly will probably be visiting the rotting cactus to lay its eggs there. They're really famous for riding on cerambycid beetles, longhorn beetles. And these longhorn beetles frequently bore into wood. And so these little pseudoscorpions can get from one tree to another, and then they'll live under the bark where they hunt little arthropod prey. A lot of times they'll be under the elytra beetles, but in this particular case, you can see them riding up on where you can actually see them. And they're tiny, tiny little pseudoscorpions. So there's no way that they could get from one tree to another unless they could ride on these beetles. Now we're going to take a little interlude of going into caves for a few minutes, because pseudoscorpions are a really good way to look at cave organisms. And this is a pseudoscorpion that's found in our southern Arizona deserts, very commonly all over the surface at low elevations under almost any given rock that you might flip. You could find these little pseudoscorpions. This is Albirix Parvaventatus. And this is a derived species, probably descended from that surface species I just showed. And you'll notice that the pedipalps are much longer and skinnier. This one is found in only 2 places in the entire world, in 2 caves in the Rincon foothills here in southern Arizona. It's found in Colossal Cave and Arkenstone Cave, which are 2 caves that are adjacent to each other, more or less. And these long pedipalps are called a troglomorphy, which is a morphology that's associated with cave-dwelling animals. Longer, thinner legs are very common in cave-dwelling animals. The other thing that's common in cave-dwelling animals is lack of pigment and lack of eyes. And you'll notice that this cave sicurina, you can see right through the cephalothorax, right to the back of it there. And you'll also notice that it has no eyes whatsoever. So this blind sicurina is found in a single little cave in southern Arizona. But in the same cave is another Sicurina that has reduced pigment, but still some pigment, and still has its eyes. So this particular Sicurina probably hunts in the twilight zone of the cave, and the other Sicurina probably hunts in the depths of the cave. So these two species of Sicurina have evolved in a single small cave in southern Arizona. This is another little spider found in that same cave called a Darkanetta. And the closest relative to this particular spider is found in Texas, so hundreds of miles away. So this is almost certainly a unique species for that one little cave. Caves also have bat ticks like this Carrion Beetle. They could have palpigrades. They could have tiny harvestmen called Cytilcenas. This little harvestman, the body length is only about 3 millimeters long, and they have raptorial pedipalps that they capture prey with. And Pseudoscorpions species. Now, the Cytilcena that I just showed you, the little orange harvestman, and this particular scorpion are interesting because they're found in a number of southern Arizona caves, and they're considered relictual species because they're left over from a time when this whole area was more cool and moist. They were probably widespread across southern Arizona. And now that for the past roughly 11,000 years, the climate has gradually grown more hot and dry, and it has stranded populations of these little harvestmen and these scorpions in caves and up at the higher elevations on mountaintops. So you have isolated populations in what are called the Sky Island Mountains, and also in caves in low elevations where they get a little extra protection from desiccation in the caves. Another species of cave spider found in the particular cave where I showed that the Cytilcena and most of those other animals were found. This is Loxosceles sabina. It's a cousin of the notorious brown recluse spider, which is Loxosceles reclusa, found more or less in your neck of the woods. Loxosceles sabina, though, has been found in only one cave, this one little cave, and you can see it captured one of those Pseudoscorpions scorpions in the cave. Loxosceles arizonica is also found in quite a few southern Arizona caves, as well as in our homes, which are artificial caves. And of course, these have the same venom that the brown recluse has, which is called sphingomyelinase D, which basically liquefies tissue. So when we're messing around in caves or in our garages, you have to be careful to wear gloves or just be aware of the fact that these very shy reclusive spiders could be hiding somewhere in there. Another spider that is frequently found in caves are spitting spiders. Now, this is a spitting spider, and what they have is a high domed cephalothorax, which accommodates a huge glue gland as well as a venom gland. So they capture their prey by squirting out two streams. Here, there's a little stream here and a little stream there. And when it's squirted out, it's liquid, but almost as soon as it hits the target, which is this cricket, it starts to contract. And it actually is a mixture of glue and silk, which this spitting spider is spitting out. So here's another shot of one in the act of spitting. And to give you a close-up of that view, so these are the tiny little fangs and the actual streams of the glue and silk being squirted out. Now, the entire spitting sequence takes only 25 milliseconds, which is 1/40th of a second. And you will observe that as the spider was chasing this cricket, the glue and silk left these moist lines on this rock here that look almost like the barcode at the grocery store. So what this indicates to us is that these the fangs are actually oscillating back and forth very rapidly. And they've actually been measured at oscillating at the rate of roughly, well, on average, 850 hertz, which is 850 full back and forth cycles per second. So the spitting sequence only takes a 40th of a second, and the fangs are oscillating back and forth very, very rapidly. So how do they achieve this? Well, they have tiny little fangs. So here's an electron micrograph of the actual fang, and this is a little bit closer up. And you have a scale down here. From this point here to this point here is 100 microns. And I measured this fang as being only 70 microns long, which is the average width of a human hair. So the length of the fang is only as long as a human hair. And you notice that this fang, instead of being hollow with a hole at the tip, it has an open tapering funnel shape in order to direct the stream of glue and silk. After the cricket or other prey is securely fastened to the substrate, then the spitting spider can go and do the killing bite, which it has to do in a very thin part of the cuticle because those fangs are so tiny. Then it cleans off its feet, and then it carries off its prey to feed. So what supports all those various hunters in the cave, including the spitting spiders and all those other little scorpions and harvestmen and whatnot? Well, first of all, you have camel crickets. They're a common cave inhabitant, and they are favorite prey of some of the larger predators. And you also have sosids, which are known as book lice. Now, book lice are really cool because what they do is they feed on fungus. The fungus grows on bat guano, and then the various little pseudoscorpions and small predators feed on the sosids. And so without the bat guano, you wouldn't have much of an ecology in that cave at all, because the whole ecosystem is based on some of the resources coming in the form of bat guano and supporting these little sosids, and then those in turn support all kinds of little predators. So we've talked about how a cave, one single cave, can support quite a population of predators, and that you can have evolution taking place in that one little cave, you know, producing blind cave sicurinas and eyed sicurinas and all sorts of things. And yet that cave is connected to the outside world by way of the bats bringing in the bat guano. Okay, so it doesn't operate in isolation, even though the gene pools of some of those animals are isolated. Now let's look at an even smaller system, which is a single black widow web. Now, most of you know what a black widow looks like. And this is a Western black widow out in a natural setting where there's a rock overhang. And what you'll notice is that she has an egg sac that she's incubating up here. Now, this is a sort of it's hard to see here, but she's got a tubular silk retreat that she's keeping her current egg sac in. And the egg sacs where babies have already emerged have been discarded. They've been pushed outside of the retreat, and she's keeping her precious little egg sac right up there. And she's feeding on a grasshopper she had captured. So here's a black widow web. And you'll notice, first of all, that there's two egg sacs out at the periphery of the web, which is a really strange place for a black widow to keep her egg sacs. Normally, she keeps them up in her retreat where she can guard it very easily. And you'll also notice that there are 4 little other spiders hanging in this web. There's one here, one here, one here, and one here. And if you look more closely at those spiders, these are called Argyrodes Pluto. And these are kleptoparasitic spiders that are obligate kleptoparasites in the webs of black widows. They always live in the webs of black widows, and they're tiny little spiders. And some of the time, what they do is they glean small prey that the black widow ignores. For example, here's a little tiny moth that this little Argyrodes Pluto is feeding on in a black widow web. But more often, what you see them doing is they have a black widow egg sac out that they have stolen. And in this case, these 3 Argyrodes Pluto, these 3 adult Argyrodes Pluto, there's a female here, an adult male here. And you can see that this adult male is feeding on a baby black widow here. Our Western black widows, when they're babies, they're actually pure white. So he's feeding on a little baby black widow. And when he was done with that, he went and tried to extract a second one. He went over to the little hole. You can see the empty eggshells from the black widow babies that these spiders were pulling out the eggshells as they were trying to grab a baby spider in there. What they do is they reach in with their chelicerae and they extract one baby spider at a time and feed on it. So here's one of the little female Argyrodes feeding on a baby black widow that she has extracted from that same egg sac. And they fed from there over several days, taking turns. So they share in the booty, if you will. When they steal a black widow egg sac, any one Argyrodes that steals it shares it with all the other adult Argyrodes that live in that web. And they all feed from that egg sac. So the thing that's really strange is that most Argyrodes live in the webs of large orb weavers, like Argiope. And the big orb weavers wrap up prey and they store prey in their web. And most Argyrodes around the world, what they do is they're kleptoparasites that they steal the prey of the orb weavers and they feed on the prey. So our black widow Argyrodes stealing the egg sacs is really different. What they've done is they've switched over from stealing prey in a orb weaver's web to stealing egg sacs from a black widow. And why is that? Well, first of all, the Argiope in our area in southern Arizona, when they get ready to lay an egg sac, they go off to a secluded place, they produce one, and they guard it, and then they die. So there's just one egg sac at a time, usually, with these Argiope. We have Neoscona Oaxacans. These are fairly common large orb weavers. Why would the Argyrodes not use them? Well, these take down their webs almost every day, and they're only really large and abundant during the summer monsoons, which only gives the Argyrodes about 8 weeks in which to do their entire life cycle on, more or less. So these wouldn't be a very good target. Metapyra is around almost all year round, but these are not terribly big orb weavers. And when they produce egg sacs, these are the egg sacs of this little Metapyra, and you can see that they string them up with lots and lots of silk. So a relatively small orb weaver probably wouldn't be able to capture enough prey for the Argyrodes to support itself with. And if the Argyrodes tried to steal these egg sacs, it would probably be quite difficult. So instead, they steal black widow egg sacs and feed on baby black widows. Here's a close-up of an Argyrodes feeding on a baby black widow. And one of the things that's really interesting is that because they only make a small hole in the egg sac, it means that it doesn't compromise the development of the remaining baby black widows in that egg sac. And so baby black widows eventually emerge from the egg sac, the remaining ones. And this particular egg sac, I counted over 70 baby black widows that successfully emerged from it, even though these little Argyrodes had been feeding from that egg sac for several days. So by extracting just one baby black widow at a time, it still leaves quite a few, which is a sign that possibly these have undergone some fine tuning as a parasite in the webs of the black widow. By not killing off all the baby black widows, they guarantee that the next generation of Argyrodes will have black widow hosts to to roost in. Now, one of the things that's kind of interesting is that these black widows, when they emerge from their egg sac, they hit the web running. They're literally racing across the silk of the black widow web, and they disperse off of that web completely in about 2 hours. Now, I did see an Argyrodes briefly chase a group of tiny baby black widows, but they quickly outran the Argyrodes and were able to escape off. But I call this precocious dispersal because compared to the green lynx spiders, where you saw the babies hang around on the egg sac for several days and undergo a molt and all that, these are already ready to go. They hit that web running, they're out of there. And they don't hang around at all on that maternal web. So I kind of think that this might be a response to the presence of the kleptoparasitic predaceous little Argyrodes in the web of the black widow. So what triggers a stealing incident? Well, the black widow has to be away from her silk retreat by several, roughly 20 centimeters before the Argyrodes will even try to steal the egg sac. And she has to be wrapping prey. That's what triggers a stealing event. At that point, the little Argyrodes goes up to the silk retreat. She cuts down, and I mean, she'll rush up there. She'll go as fast as she can go. And she'll zip into that silk retreat. And a few minutes later, she's coming out with the egg sac of the black widow. Then she moves it off to one side of the entryway. And here she is pulling it off to one side. This is because she needs to suspend that egg sac away from where the black widow might find it. And she seems to know right where to put this egg sac in the web where the black widow very rarely goes. If the black widow happens to encounter the stolen egg sac, she will carry it back to her retreat. And in one case, we had a monsoon storm where the black widow web was damaged by wind and rain. And so the black widow came out and she was doing all kinds of repair work on her web, and she encountered a stolen egg sac out at the periphery of her web. It had a little dent in it from the storm, so it was easy to recognize, and she immediately carried it back to her retreat. Well, for the next 3 nights, the Argyrodes took turns trying to steal that egg sac again. And on the 3rd night, they succeeded. And I was able to recognize that same egg sac because it had the little dent in it. So it's remarkable how the poor black widow will try to retrieve her egg sacs, and then the Argyrodes will try until they steal it again. So here you can see an Argyrodes suspending the egg sac, and here's some light colored objects in the background. And what those are Argyrodes egg sacs. So she lives in the web of the black widow, but she has a little spot that she's set up that seems that the black widow hardly ever goes to. And that's where she makes her own egg sacs, and that's where she carries off the egg sac of the stolen from the black widow. Now, after she's fed on that stolen egg sac for a while, what she'll do is become very fat, and she's ready to make her own egg sac. So she starts out making a little goblet shape, and then she tips it on its side, and she starts to expel eggs in there. And she does it in one continuous push. It takes her about 1 minute to push out all these eggs. And you can see this deep dimple forming in her abdomen as she's pushing out her eggs. And here she is, she's just finished pushing them out. So you can see the volume of eggs that this tiny little spider has pushed out in the space of about a minute. And then she puts flocculent silk on there. She wraps the whole outside with flocculent silk, and then she wraps it with very fine silk and makes a little neck at the bottom. She spends about 4 hours making this egg sac, putting layer upon layer upon layer of fine silk on there. And at the end of it, she rests and guards it. And she stretches these 2 long front legs across it. And you'll see in a little bit why she guards it. So she's resting there with the egg sac just suspended in the black widow web and guards her egg sac. So when she starts out, before laying eggs, she looks like a little sparkly Christmas tree ornament. But by the time she's finished, she looks kind of shriveled and emaciated. And at that point in time, she's ready to go steal another black widow egg sac. So these things go cycle after cycle. She would steal a black widow egg sac, they'd feed on it for about 10 days, and then she'd be ready to produce her own egg sac, and then she'd be ready to steal another black widow egg sac. So fortunately, black widows can produce multiple egg sacs. This particular black widow was in a web that was on a bicycle that I had parked under a porch light. So this was a really ideal situation. It was right outside my front door, and the porch light gave me nice dim light so that I could actually focus and observe things without disturbing the animals in any way whatsoever. They were used to the porch light, and the light, it was a yellow bug light, but it drew in a fair number of insects. So the black widow did very well. And she produced a total of 9 egg sacs over that particular summer. And of those 9, the Argyrodes managed to steal 7 of them. So that'll give you an idea of why they might have switched to something like a black widow compared to the orb weavers, because the orb weavers, there's no way in heck that they could produce 7 egg sacs in one summer season. You know, so this is a continuous supply of food for these little Argyrodes. Now you'll notice the entryway of the retreat here of the black widow and the tire here. So you can see that it's my bicycle parked there. And here she is carrying off something. And you'll notice a little black something on the bottom there. We'll get back to that. Now, whenever the black widow was out wrapping prey in her web, she would stop and rush back at some point to her silk retreat, and she would check in there. And I think that she was checking on her egg sacs. But in this case, she got back to the silk retreat after the little Argyrodes had already transported the egg sac just far enough away that the black widow did not seem aware of where this was. Of course, black widows they use their sense of motion in the web more than their eyesight, okay? So even though this little thing was just out this far, the black widow did not seem aware of it. By morning, here's where the silk retreat was, up on top of the bicycle tire. And here's where the little Argyrodes had carried this new egg sac. And there she is feeding on a freshly extracted baby black widow. So I mentioned a little black something on the egg sac. Well, there it is, a better view of it. So here's the Argyrodes carrying off this egg sac, and here's something else on it. And this something else turns out to be Philolema latrodecti. Latrodecti is the same, is named after the genus of black widow, which is Latrodectus. Okay, so Latrodectus is black widow. Latrodecti refers to the fact that this wasp is an egg parasitoid wasp that specializes on black widow eggs. So sometimes what happens is that instead of hatching out a bunch of baby black widows, what you get are a bunch of little wasps from an egg sac. That's a closer view. But they have competition. So here's a little fly called Pseudoscorpions signatus. And what these flies do is that they look for a black widow egg sac. They'll deposit their eggs on the outside of the egg sac. Their maggots will tunnel through the silk and will feed on the eggs of the black widow. So there are 2 competitors in this very same black widow web trying to get those eggs in addition to the Argyrodes. But the Argyrodes has problems of her own because she has to defend against this little Arachnids dazzes. This is a little egg parasitoid wasp, and she's trying to shoo this wasp away. And this wasp kept flying around. Sometimes she would grab the wasp and fling it. Here she is actually flinging the wasp. And then sometimes she'd have to chase the wasp around before she could catch it. Here she is chasing it frantically around. And she'd always be waving her legs at it, trying to shoo it off the egg sac. So this little wasp was trying to lay eggs on this egg sac so that its larva could parasitize the Argyrodes eggs. So it gets very complicated. After about 40 minutes of watching this frantic little mother trying to defend her egg sac, I finally caught the little wasp so that I could get it identified. And as a final note, in terms of some of the predators in that black widow web, I removed a total of 7 pirate spiders from the black widow web over the course of the summer. Pirate spiders feed on comb-footed spiders, including black widows, and also feed on orb weavers. So they'll infiltrate a web and actually kill the resident spider and feed on that. So I captured 7 of these pirate spiders over the course of a summer. I wish in a way I had left them to find out who they were going to preferentially pick off, because if they had picked off the Argyrodes, it would have actually benefited the black widow. But if they had killed the black widow, it would have been a disaster for my whole little study. So I was capturing these and removing them as I found them. So now we're going to look at one other pair of kleptoparasites and comb-footed spiders. This is Tedaren Sisophoides, which is another cousin of the black widow. They're in the same group of comb-footed spiders. We find them in Arizona, usually along riparian areas. So these are the leaves of cottonwood. So it's along a little stream out in the desert with cottonwood trees. And what she's done is fasten these together. And sometimes they use a single leaf that they sort of tie into a curl shape. And if you twisted that leaf around, here's the Tedaren Sisophoides with her egg sac. And this is where they use as a retreat. And they guard their egg sacs in the leaf retreat. But you'll notice that there's a lot of silk here. So Argyrodes would have a pretty tough time trying to get these egg sacs. Now, living in the web of the Tedaren Sisophoides, I found these kleptoparasitic spiders. A relative of Argyrodes, this is called Neoscona Oaxacans. It's a female with her own little egg sac, which looks a lot like the Argyrodes egg sac, but you'll notice that there's some debris always fastened on the outside of these ones. And at rest, these spiders look like a little bit of debris hanging in the web. The females are highly variable in color. Here's a female. And the males have odd little protuberances and bumps on their heads. Here's a little protuberance and a little bump. But the reason why I'm showing this male is you'll notice that on the far side of his body, he has these 2 long legs, this leg here and this leg here. But on the near side of his body, both of those legs are gone. They're just little stumps. So put that aside in the back of your mind. I'll come back to that a little bit later. So what Neoscona had been previously documented as living on are these small spiders called Philipinella. These spiders lack venom. They're in the only family of spiders that completely lacks venom. And they are facultatively communal. So they form little colonies where the webs are adjacent to each other and above and below each other. So there's a big supply of little spiders and they're completely defenseless. So those Neoscona, what they normally have been documented as doing is going and killing and eating the individual little Philipinellas. However, in the webs of Tedaren Sisophoides, I found that they were living as kleptoparasites, gleaning prey from the Tedaren webs, very much as if they were kleptoparasites in an orb weaver's web. So here's a little juvenile Neoscona feeding on an ant that has gleaned in the web of a Tedaren. Also in the Tedaren webs, guess who showed up was Philolema latrodecti, an egg parasitoid wasp. Now, one other interesting thing is you'll notice off to the side of this Tedaren refuge, there's a little odd spindle-shaped cocoon. And if you look closely at this cocoon, and if you were to raise up the animal that comes out of that cocoon, it's an ichneummonid wasp. So in these Tedaren webs, there were ignomonid wasps. Now what these Zatypoda do is that they are parasitoid wasps on comb-footed spiders. So on things like the Tedaren and on the Neospintharis. So here's a Zatypoda wasp larva on a spider. And what they do is they feed on the spider until the wasp larva is ready to pupate, and then they kill the spider and they spin their cocoon. So here's a Latrodectus Hesperus web in the same area that the Tedaren webs were. You can see these cottonwood leaves. So this web was right next door to a Tedaren web. So the Tedaren Sisophoides and the Latrodectus are sharing the same habitat. And as reference, this is the front of her silk little tunnel refuge. And there's a bent cottonwood leaf right here. See how this leaf has a sort of a bend in it? Okay, use that as a reference. This is the same leaf in the background here, okay? But I'm gonna focus a little bit in front of it. And what do you see where these white arrows are pointing? We have Neospintharis, 5 of them right here, one up here, one here, one here, one here, and one here. We have Neospintharis bavokiveri also living in the black widow web, okay? And one more animal living in that black widow web, one lone little Argyrodes Pluto was living in the black widow web, attempting to guard her little egg sac, but you will notice that she's missing her 2 long front legs. She has only 6 legs, and the 2 really long ones are both missing. And I think it's because of the Neospintharis also in her web that shows some degree of aggression, shall we say, since a male Neospintharis was missing 2 legs, and here's a little Argyrodes Pluto missing a couple legs. And I never saw any Argyrodes Pluto missing legs if Neospintharis were absent. So my suspicion is that it's aggressive interactions between the kleptoparasitic spiders in this web that led to her missing her legs. So let's just briefly review what a single black widow web might have. It could have Argyrodes Pluto, 1 or several of those. It could have Neospintharis bavokiveri. It could have Philolema latrodecti. You could have these flies that are also egg predators. You could have this little egg parasitoid wasp that also goes after the egg sacs of the Argyrodes Pluto. You could have the Zatypoda wasps. And you could have the mimetis as pirate spiders coming into the web and going after any of those other spiders. So it's a very complicated situation. And one little web could have several trophic levels and highly complex interactions. And what this brings me to conclude with is the idea of Indra's net. So Indra's net is a metaphor that came out of 3rd century India. And what Indra's net was a spider web where every strand of the web connected the entire universe, and the web was hung with dewdrops, and every dewdrop reflected the entire universe. And in our modern day society, it's interesting because we use the same metaphor almost exactly. We use the World Wide Web and the internet as our means of staying connected among our whole world. So I think it's interesting that it's sort of like history repeats itself in some ways. And so I want to finish with the idea that we're all interconnected, that even if sometimes we're in our own little bubbles or in some way feeling isolated, we are still part of the larger picture, the universe, just as those little cave organisms are still connected to the outside world and we're connected to our entire planet, and we need to take good care of it. So if any of you are interested in learning more about arachnids, I do have this book out by Princeton University Press, published in 2018. And I'm ready for any questions now.
Jillian, that was amazing. Thank you. Can you unshare so we can come back together as a group? Whoa, I feel like I have gone to a whole nother universe and seen a whole secret world going on. I'm so excited to have learned all of this. If you have a question for Jillian, please raise your hand, and or you can put it in the chat box if you want to do it that way. Raise your hand, and I'll recognize you and unmute you for a question.
Oh, okay. Good.
Janina, go ahead. You have to unmute. You have to unmute first.
Good, you didn't hear that. This sounds like a stupid question, but does the female black widow have to mate every time to get one of those 9 egg sacs per season?
Nope. Nope. Okay. She can store. Yeah, she can store the sperm. So, so after mating with a single male, she can actually store sperm for quite some time, and they can actually live for several years. A female black widow can live for 3 years. So during that time, so you could imagine how that would be a wonderful resource for something like these little kleptoparasitic spiders to be able to continue to tap into that wonderful resource that continues to produce these wonderful egg sacs, you know.
So one male spider could last out like the whole 3 years of the black widow's life?
She only has to mate once, and then he tends not to live very long. Male spiders tend not to live very long, and your Western black widows actually very, very rarely kill the male spider. They'd have to be really hungry to kill the male spider. A lot of times the male black widows hang around in the female's webs for a period of a week or more, and they sometimes even steal a little bit of food from her and almost live like kleptoparasites themselves. So they're much smaller than the female, and I've seen as many as 2 or 3 males hanging out in a female's web. Yeah.
Thank you.
Any more questions?
Mary has a question. She asks, besides food for other parasites, what environmental advantages do black widow spiders provide?
What other advantages? What was the question?
Besides food for other parasites, I guess for other animals, what other environmental advantages do black widow spiders provide?
Oh, what do they provide? Okay, well, they're just a good apex predator, I would say, in terms of arthropods, you know. Of course, there's other animals that might feed on black widows as well, you know, such as, you know, some of the vertebrates might feed on a black widow. But yeah, they're just part of the ecosystem in the sense that you do need to have a balance of predators so that something's out there to eat all the crickets and other little herbivores that are out there. So they're just part of a balanced ecosystem, I would say.
Jillian, I think Jim asked a question that I think everybody's interested in learning about: how you take those macro photographs. What gear do you use? The techniques. I mean, we're looking at such small little things, like a blade of grass. How do you know where to go, where to look? What do you use?
Okay, I use a Canon camera. I've actually gone through. I'm on my 3rd Canon camera body. I started out with an EOS Rebel, and I'm on my 3rd EOS kind of body. I use a variety of different macro lenses, including one that's called the MPE 65mm lens, which is only Canon makes, and it's kind of a super macro lens that goes from 1x to 5x. So that's very, very helpful for photographing small things like these Argyrodes. And I use a Sigma ring flash. Now, it's called a ring flash because it fits at the end of the lens, but it's actually no longer that annoying, nasty ring-shaped light. What they did is they put two lights that are twin lights right in the flash. And you can program it manually. And so I do all my photography with manual settings for everything. I use all manual on the camera, and I use all manual on the flash. And that allows you a lot better control when you're doing the photography. And basically, you just have to be very, very patient to get those Argyrodes photos of Argyrodes actually in the act of stealing black widow egg sacs and laying their own eggs, you know, producing an egg sac of their own. I had to stay out there all night. And the Argyrodes, for the most part, just hang there motionless in the web. And once in a while, they'll wave one leg really slowly, like they're doing Tai Chi. And I'd get all excited thinking something was going to happen, and then they just hang there for another few hours and do nothing. And I got really used to just sitting out there and learning how to sort of be alert but relaxed, because the nights really are long when they're hot, and it would be hot out there. You know, summer nights in Arizona can be 90 degrees at 11 o'clock at night, you know. And you're just sitting there in a puddle of sweat and waiting for something to happen all night long. and when they actually steal the egg sac, it takes only a few minutes. I mean, they zip up there and they've got it out in like 2 or 3 minutes flat. You know, it's not a long time. So you really need to be paying attention.
Yeah, the photographs were amazing to look at. Again, it was like we were peeking into the secret world going on and we don't even know about. And Jillian said that it took the book was 10 or 12 years in
yeah, 12 years I worked
work that goes into that. So it's pretty fascinating. Terry asks, have all spiders commonly found around humans been studied to this degree? And how can we best look up the lives of our favorites in a format a layperson can understand?
Oh, that's a really good question. Oddly enough, there's a really old book that's out there. Well, it's been around for a while, since the time I was a kid, basically. It's Among the Golden Guide series. There's Spiders and Their Kin, I believe it's called, by Herbert Levy. And it's very inexpensive, and I would highly recommend it. I still think it's an excellent book for showing some of the common spiders and giving you some natural history on them. And it's definitely geared for the lay person. So that's one of the benefits, is that the identification isn't telling you to, you know, dissect out the spider and look at it under the microscope. Instead, it's using characters that you can look at with your naked eye, you know. So that's the one I would most recommend. There will hopefully be a field guide coming out by Princeton University Press in the next few years that will be a photographic field guide of North American spiders. There's a gal working on it right now. I won't go further than that, but I'm hoping that there will be another wonderful field guide out pretty soon.
And Dave is interested, are scorpions the only arachnids that fluoresce under ultraviolet?
No. There's many arachnids that fluoresce. Actually, here's the thing about scorpions, is that they fluoresce under quite a wide wavelength of UV lights. And so some of the older UV lights, only the scorpions would fluoresce under them. If they were at like, I'm gonna say like 400 nanometers, something like that, at the longer wavelength of the upper end of what constitutes ultraviolet light, okay? As you get down to shorter wavelengths, there's a lot of arachnids that fluoresce under UV light. So there's some newer black lights available that have shorter wavelengths that are more like 360 nanometer wavelengths, and that have filters that filter out almost all the visible light. And when I have tested those against various arachnids, a lot of arachnids do fluoresce, including many of our crab spiders. A lot of the little crab spiders that are foliage hunters fluoresce very brightly, a nice bright apple green that you can pick them out from several feet away in foliage at night with the right kind of UV light. So the trick is to get one of these newer ones that have the shorter wavelength and a filter. And then you can go out at night and you can pop out quite a few arachnids that way.
Question, do you move spiders out of your home? Why or why not?
Generally speaking, I do not. In fact, I really encourage most spiders in my home because things like cellar spiders that I showed that are common in people's garages, they are fantastic in the home. I would estimate about a dozen scorpions a year get captured by cellar spiders in my house, which means that I don't step on the scorpion and get stung for every one that the cellar spiders capture. In addition to that, the cellar spiders are very good at capturing kissing bugs. And we have several species of Triatoma in our area. And Triatoma rubida, it was found that 41% of the Triatoma in our area actually do carry the trypanosome, which is responsible for Chagas disease. So I would prefer, plus my spouse is very allergic to the bites of kissing bugs, where he could actually get an anaphylactic reaction if he were bitten by a kissing bug. So I love having cellar spiders around because they capture kissing bugs. Every year, at least 1 or 2 kissing bugs are found by a cellar spider in my house. And for all I know, they might save our lives. I mean, that's really, so once in a while I capture spiders if they're like a ground hunting spider, like a Naphosid, where they've wandered in, they've gotten in somehow, and I know that there's not much prey for them. So I'll capture them very carefully and move them outdoors where they can find prey more readily. But as far as I'm concerned, things like cellar spiders and even the Loxosceles, I let them live in my house. They're shy, they're reclusive, they don't tend to attack people. And if they can capture a few kissing bugs, I figure more power to them.
And there are a lot of, going back a question, there are a lot of apps out there that you can also take photographs with your smartphone and get ID help. One of which is iNaturalist, and we actually have an online training with Bill Curtis coming up on April 21st, getting ready for this the City Nature Challenge, which is a bio blitz that happens, and Baltimore and DC and other cities across the country participate. So if you're interested in that, you can come to an online training and learn how to use iNaturalist, and then you can go out and go look closely now that we know where to look at little grass things and find all of these spiders that are hiding from us and learn where they are, what they are, and what they do. Let's see. Terry says that she has a $10 UV flashlight that she uses to find tomato hornworms.
Yes.
Could she use that to go on a spider hunt?
It depends on what the nanometer is of your UV light. They really need to be 360 nanometers or even a little less for them to pop out most of the arachnids. Now, a lot of insects do fluoresce. So, so you'll still be popping out some of those, and it's always fun to look. But as I say, it's some of the newer UV lights with a shorter wavelength that seem to be better for arachnid hunting.
All right, any other questions? We're on UV lights. Can you recommend one?
Oh gosh. I think that the best way to find them would be if you do a search online for UV lights. I got mine actually at the Gem and Mineral Show, and that normally runs in Tucson, but I don't think that that guy has his available online to everybody. But I have seen similar products available online where if you do a search, and it'll actually show pictures of some arthropods that have been taken with these shorter UV wavelength lights. They're small. They're like only about this long, I guess about 5 inches long, and they're cylindrical, about that big around. And they'll have a filter at the end of the light, and they take the equivalent of, like, I'm going to say like 2 AA batteries, something like that. And, you know, they're smallish. You can fit them in your pocket, actually. And they're amazingly powerful. I mean, I can spot a scorpion from like 30 feet away with one of those things. So I have seen online a very similar product to what I use, but you'd have to just do a quick search on UV lights for arthropods or something like that. And I think that you'll find, you know, especially ones that show examples of some arthropods actually popping out with this light. That would be the thing to look for. But see if the specifications say like 360 nanometer. That's what you want.
And Ralph says Alex has said it's been an amazing talk. I learned so much about the arachnid life cycles and ecological relationships. I echo, and I believe I echo that for everybody here, because everybody looks smarter than they did when we started. So that's great. And I, for one, am so excited. I can't, you know, spring is here on the East Coast, so it's time to get out and start exploring. Everything's coming out. So we're gonna go out and look for them, and I'm more excited about finding and learning more about arachnids around me. And now it's your homework, everybody, is to take the information that you've learned tonight and share it with others, because that's the power of education.
And I'd like to say that I've barely scratched the surface in this talk. There's so much more. So if if you dig into the subject, there's almost no end of incredible discoveries to make. And you know, nobody had known that there were any kleptoparasitic spiders that specialized on stealing egg sacs of other spiders until I happened to notice on my front porch that they were feeding on black widow egg sacs, you know? So, I mean, all around the world, people have studied kleptoparasitic spiders, and nobody had ever documented that before. So, you know, so there's lots of things to discover right on your front porch. You know, who knows what you've been walking past, and you just happen to notice it, and it turns out to be something that nobody had ever noticed before. So sometimes it just takes a little closer look and going a little slower. Sometimes that helps a lot too.
Yeah, you'll be amazed at how many scientific discoveries have been made by just folks like you and me. Remember, Darwin was an amateur scientist himself, so it wasn't a professional. So we're all encouraged to go out there and move a little slower and lower and look around. So that's great, Jillian. And you know, if there's more and you have more to share with us, we'd love to have you back later on to do some more teaching for us.
All right. Well, thank you so much for having me, and I hope that you guys weren't overloaded with all this.
No, It was great. Thank you so much. And everybody, we hope to see you again at one of our talks. We have Thursday. If you're still into little critters, remember we have Dr. Lapola from Towson University, the myrmecologist, on Thursday to talk about farmer ants and mealybugs. So that's another fascinating relationship of things that insects right beneath our eyes going on. And come back and see us. If you're interested, if you like what we're doing, become a member, and everybody stay safe and stay curious. Good night, everybody.
All right. Thank you so much. You guys take care. Bye-bye.
Thank you.